Assalamu alaikum. Hello everyone. Let us discuss about the virus severe acute respiratory syndromes, also called as SARS virus, the general properties, structure, transmission, replications, and the pathogenesis, how to diagnose this virus, preventive measures, and treatment. What is SARS virus? SARS or severe acute respiratory syndromes is highly contagious respiratory illness caused by the SARS-CoV virus. It can get serious, causing severe pneumonia and even respiratory failure. Classifications of the SARS virus: order Nidovirus and family Coronaviridae, genus Beta coronavirus, species severe acute respiratory syndromes and related to coronavirus. Structure of SARS virus. SARS virus is spherical shape with distinctive spike protein producing from its surface a lipid envelope. The virus is surrounded by a lipid envelope derived from the host cell membrane, which is stuffed with viral spike protein. This spike protein facilitates the virus entry into the host cell. Inside the virus, there is a single standard RNA genome surrounded by a lipid envelope. The genome encodes various proteins necessary for viral replications and assembly. Nucleocapsid bound to RNA genome forms nucleocapsid. Central organizer of COVID assembly determine the shape of viral envelope is membrane protein. Envelope protein interacts with membrane protein to form viral envelope. The structural components and genetic material collectively enables the SARS virus to infect and hijack host cell during an infection. Mode of transmission. Virus can spread through tiny droplets and aerosols in the air, especially in enclosed spaces. Close contact spread. The virus can also spread through close contact like touching or being near uninfected persons. Fomite transmission. The virus can land on surface and objects, making them potential source of infection. Replications of SARS virus. First step is virus entry. SARS coronavirus enter host cell through endosomes or plasma membrane fusions, spike protein and ACE2 interactions. Spike protein S1 and S2 attaches to the host cell membrane through ACE2 receptor. Griffin inhibits viral entry by binding to the spike glycoprotein. Exogenous heparin competes with heparin sulfate, hindering S protein binding. Cell surface co-receptor and enhancement. Cell surface vimentin is a crucial co-receptor for ACE2 binding, and heparin sulfate enhance ACE2 binding by binding to the receptor binding domain. Endosomal entry activations. Cathepsin L activates the spike protein in endosomes, while lysosomotropic agents like baphyllomycin A1 or ammonia chloride block pH dependent cysteine protease, inhibiting viral entry. Plasma membrane fusions. Transmembrane protease serine 2. A cellular serine protease cleaves the spike protein between S1 and S2 domains, initiating fusions of the viral membranes with the plasma membrane for efficient replications. Antiviral immunity considerations. Plasma membrane fusion entry is less likely to trigger host cell antiviral immunity, enhancing the efficiency of viral replications. Second and third step is translations of viral replication machinery and replications. Viral RNA is released into the host cell, initiating the translations of polyoproteins. Coronavirus genomic RNA encodes non-structural proteins and structural protein, with polyoproteins PPLA and PPLAB being initially translated. These polyoproteins are cleaved by papain like protease and 3C like protease to form functional non-structural proteins. Crucial players like helicase and RDRP in functional to non-structural proteins play a vital role in viral RNA synthesis. RDRP, a key enzyme, is target for inhibitors such as favipiravir and penciclovir. Additionally, the replications of viral RNA can be hindered by kinase signaling pathway inhibitors like saracatinib. Resveratrol, known as inhibitor 10, can decrease the expressions of the N protein, the non-structural protein host shut-off factor on early protein in translations, disrupt host mRNA, speeding up its degradations. These disruptions aid in the virus in avoiding the host immune response. Translations of virus structural proteins and virion assembly. The viral replication process involves the orchestrations of structural protein RNA by OGRP. Structural protein is envelope and membrane proteins are produced by ribosomes on the endoplasmic reticulum. Viral RNA replication takes place within the double membrane vesicles formed by the endoplasmic reticulum, providing protection against the host immune systems. Non-structural protein 3 creates pores, allowing the viral RNA to exit double membrane vesicles for virion assembly. Nucleocapsid proteins are assembled in the cytoplasm from genomic RNA. These nucleocapsid merge with the virion precursor and are transported from the endoplasmic reticulum through the Golgi apparatus. Small vesicles add in the transporting the assembly to the cell surface. This well coordinated process results in the assembly of mature virions poised for release and release of virus. Virion are released from infected cell via exocytosis to search for another host cell. Oseltamivir, a medication stops this release by blocking neuraminidase, which is crucial for this process. SARS coronavirus 2, the virus causing COVID-19 stand out because it has an additional cutting sites in its spike protein compared to other coronavirus. This cutting process involves furin at the S1 and S2 site, likely happening during virion release through cell compartments like Golgi apparatus or lysosomes. This prepares the spike proteins for a second cut at the S2 site by transmembrane protease serine 2. Changes or mutations in the S2 site are linked to notable SARS coronavirus 2 variants alpha, beta, delta, omicron. These mutations can impact the virus behavior and contribute to the emergence of new variants of concerns. Pathogenesis of SARS virus. SARS coronavirus virion consists of spike, nucleocapsid, membrane, envelope protein. Infection stage like attachment, cleavage, activation, fusion. Stage 1 attachment. S protein binds to ACE2 receptor on the host cell. Stage 2 transmembrane protease serine 2 cleaves the S protein. Stage 3 activation. Cleavage activates the S2 domain for fusion. Stage 4 activated S2 fuses viral and host lipid bilayer, depositing the viral RNA genome into the host cell. Viral replication. During the replication, SARS virus produces double standard RNA intermediates. The immune system detects double standard RNA through melanoma differentiation associated protein 5 or retinoic acid inducible gene 1, initiating a signaling cascade via mitochondrial antiviral signaling. This cascade triggers the productions of type 1 and type 3 interferons, which act locally and systemically. Interferons bind to cell surface receptor, activating the Janus kinase and signal transducer and activators of transcriptions of 1 by 2 signaling cascade. This cascade results in the productions of interferon stimulated genes with antiviral functions. Specific structure: double membrane vesicles involved in viral replications, interferon sensitive response. Clinical signs and symptoms. Incubation period is 2 to 10 days. Complications such as pneumonia, acute respiratory distress syndromes, acute cardiac injury, damage to the heart muscles, acute renal failure, a sudden loss of kidney function, encephalitis, inflammations of the brain, hypotension, abnormally low blood pressures, early symptoms, fever, headache, muscle aches, respiratory symptoms, and late symptoms such as dry cough, shortness of breath, difficulty in breathing. Diagnosis of SARS virus, nucleic acid testing, RT-PCR, reverse transcription polymerase chain reactions, detect viral RNA, real-time RT-PCR, allow quantifications of viral load, serological test, detect antibodies IgM, IgG against SARS virus, ELISA and rapid test are common methods, immunofluorescence assay. Treatment and preventive measures. Treatment. Severe cases require intensive supports. Ribavirin, lopinavir, ritonavir, systemic corticosteroids. Preventions and control measure. To prevent the spread of the disease, wear a mask, practice hand hygiene, and avoid contact with infected individuals. If sick or exposed, isolate, limit large gathering, and travel to reduce the risk. Implement rapid testing and contact tracing to identify and control the spread of the virus. Provide necessary medical care and resources to manage the Thank you everyone for watching my video. I have attached reference in this video.